Welcome back friends, na wote ambao mnafuatia Dr. Mlelwa Lectures. Kwenye kipindi kilichopita tulikuwa tunaendelea na kusolve maswali ya advanced biology na naelezea jinsi ambavyo tunaweza kujibu maswali mbalimbali. Leo tunaendelea na swali la 5 katika pepa ya Biology Giants. Swali la 5A walisema state the role played by each of the following chemical substances in light independent reaction of photosynthesis. Na kwa mfano hapo alisema number 1 ATP, wakasema waandike 2 points. Rubip kusema waandike point ngapi? Probably ni moja. NADP H2 which they reduced in NADP, carbon dioxide and rubisco. Kwa hiyo kwa nature swali kama inaonyesha kabisa kwamba hizi ambazo haukutajiwa is 1 point and hii hapa ambayo imetajiwa it's 2 points. And then part B, discuss how come and DC4 photosynthesis improve upon C3 photosynthesis, include both advantages and disadvantages of processes, describe appropriate leaf anatomy to complete your discussion. Now, question A, it is simply of mentioning the laws of the substances you are given, and that is the knowledge from anatomy. I mean the knowledge from nutrition. Sometime na mix in anatomy kwa sababu huwa nafanya vitu vingi, so wanaweza nikamix vya huku chemistry, nikamix na biology, au nikamix na vya medicine. Anatomy hiyo ni course ya chuoni first year. Anyway, ukifika chuoni first year you can enjoy it, you can enjoy anatomy. So the rules in photosynthesis, first we can start with ATP. ATP, it has 2 rules in photosynthesis. Now, more especially these rules, they have been explained well by the biological science book. Moja kati ya kitu ambacho natakiwa uongezeke sana ni uongezeke uwezo wako wa kutumia vitabu. Uwezo wako wa kuweza kusoma kitabu na kuelewa mwenyewe bila kufafanua namba. That's very important ability you need to develop, simply because that will enable you to search for the answers in the books without being given the answers by a teacher. Kwa hiyo rules of ATP in photosynthesis. Kwanza, ATP is used to reduce phosphoglyceric acid, which is PGA, into phosphoglyceraldehyde, which is PGAL. Phosphoglyceric acid is the 3-carbon acid. It contains an acidic carboxylic group. PGAL is a 3-carbon sugar and it contains an aldehyde group. Thus, ATP and the reduced NADP are used to remove 1 oxygen from phosphoglyceric acid. That's the reduction. And the second role of ATP in photosynthesis is that is used in the regeneration of carbon dioxide acceptor. In the carbon cycle, some of the PGAL is used to regenerate or convert ribulose phosphate into ribulose biphosphate. That means ATP is taken from, phosphate is taken from ATP and it's added to the ribulose phosphate to make ribulose biphosphate. The second is the role of ribulose biphosphate in the dark reaction of photosynthesis. As we know, ribulose biphosphate is the carbon dioxide acceptor molecule which occur in the stroma of chloroplast. Addition of carbon dioxide to rubip form the temporary unstable 6-carbon product which break immediately into 2 molecules of 3-carbonic compound glycerate phosphate or phosphoglycerate. These are just additional informations regarding the role of rubip in photosynthesis. Then the role of NADPH, NADPH or NADPH2, that's reduced NAD. Reduced NAD is used in reduction of glycate phosphate. The first product of photosynthesis in C3 plant into triose phosphate, which is the first carbohydrate to be formed in photosynthesis of C3 plant. So it performs this work with the use of energy from ATP. Now, one thing you need to remember in this part that sometimes you'll be asked the rule of NADP, not NADPH. These are two different substances. However, their functions can seem to be closely related. Actually, the NADP is the oxidized NAD form. NADP is oxidized form. And this NADP, its role, the role of NADP, it acts as the carrier of proton. You can say it the proton carrier or the hydrogen carrier from light reaction to dark reaction of photosynthesis. In dark reaction, reduced NADP is used to reduce phosphoglyceric acid to phosphoglyceraldehyde, which is the first carbohydrate to be formed during photosynthesis. As per the equation above, actually here there is no equation. You can assist these answers with equation. If you are able to write the equations, it will be more relevant and it will increase your chance of performing well. Carbon dioxide is used as the source of carbon and oxygen used in making carbohydrate during photosynthesis. Carbon dioxide is used as the source of carbon and oxygen used in making carbohydrate during photosynthesis. So since carbohydrate is made up of carbon, hydrogen, and oxygen, the carbon and oxygen comes from carbon dioxide and hydrogen comes from water. Rubisco is an enzyme which catalyzes the carboxylation of rubib to form a temporary 6-carbon compound which break into the 2 molecules of 3-carbon compound glycerate phosphate. Rubisco is sometimes called rubib carboxylase. Catalyzes the acceptance of carbon dioxide by ribulose biphosphate during carbon cycle as per the reaction in 5A3 above. Actually, as I told you that there is no reaction written here, but hopefully you know these reactions which are involved in the conversions of these compounds. Now, note that students should also know the rules of water and light in photosynthesis. All of these, they are different questions which can be asked in the exam. So Latano B, discuss how C4 and the CAM photosynthesis improves upon C3 and photosynthesis. Include both advantages and disadvantages of process. Describe a property, leaf anatomy to compare discussion. Now, let me find you the answers for this question. Okay, now we can continue this question number 5B. Moja kati ya kitu ambacho tujue ni kwamba marking guide huwa haiwezi kuandika kila maelezo. Yaani hii swali kwa mfano ni ref. Unaona, discuss how C4 and the CAM plant photosynthesis improve upon C3 photosynthesis. Unaweza ukaelewa kitu kimoja kwamba kwa swali kama hili unatakiwa uelezee biochemical pathway za C4 na CAM and how these pathway provide an advantage to C4 plants. Kwa hiyo kivipi C4 plant na CAM plant zina advantage ukilinganisha na C3 plant? Kwanza tukianza na C4 plant. C4 plants, it has 2 advantages. Kwanza, it minimizes photorespiration and reduce water loss through leaves. Hiyo kama umesoma nutrition kwangu tulielezea sana. Advantage of the C4 plants over C3 plants. Unaona? Kwa hiyo tunaposema ina reduce photorespiration, hapo natakiwa ueleze kwamba inakuaje kwenye C3 plant kunakuwa kuna photorespiration, alafu kwenye C4 plant kunakuwa hakuna. Na kwa nini tunasema ina reduce water loss through leaves? What happens kinachopelekea water loss through leaves iwe inapungua? Unaona, yani water loss through leaves iwe sio nyingi kama vile ambavyo itegemewa. Kwa hiyo hizo ndo advantage kubwa mbili za C4. Lakini pia plant naye advantage yake kubwa ni ya kusaidia kuprevent water loss through leaves. For discuss the temporary separation of carbon dioxide. How carbon dioxide in the form of malic acid is transported to the vacuole during night, and how it is later transported to the chloroplast during day, because carbon dioxide is taken in at night, stomata are closed during the day, and the result water is reduced, especially in hot climate. For both CAM and C4, a disadvantage is that these pathways require additional energy. Hapa kiufupi ni kama vile unahitajika kuelezea mechanism, lakini uelezee mechanism hasa kwa namna ya kueleza advantage na disadvantage za hizi mechanism. Question number 6A, kwanza kuna definition kama limiting factor of photosynthesis, definition of photorespiration, definition ya C3 plants, C4 pamoja na Kranz anatomy. Vitu kama limiting factors of photosynthesis ni vitu ambavyo walimu wengi hawavifundishi. Sasa ndio maana tunakuwa tunapata ugumu sana kusoma masuala ya biology kama mtu hajasoma concept kwanza na hajui. Limiting factor of photosynthesis is that factor that directly affect photosynthesis process if its quantity is changed or is sometimes present in the environment in such short supply that it restricts photosynthesis process. Three factors can limit the speed of photosynthesis. These are light intensity, carbon dioxide concentration, and temperature. Unaweza kuona how light intensity is a limiting factor, carbon dioxide concentration is a limiting factor, and the rate of photosynthesis, how it is the limiting factor of photosynthesis. Kiufupi, limiting factor ni ile factor ambayo ukipunguza itapunguza speed ya photosynthesis moja kwa moja. Photorespiration is the metabolic pathway occurring in plants. It's the metabolic pathway occurring in plants in the presence of light, which uses oxygen and produce carbon dioxide and reduce the photosynthetic output in these plants. So ni metabolic pathway ambayo inatumia oxygen na ina consume kile ambacho kilitengenezwa, or the process of uptake of oxygen and production of carbon dioxide in light by the photosynthetic tissue. It is also called oxidative photosynthetic carbon cycle, OC2 photosynthesis, na diagram yake ni hiyo hapo chini. C3 plants are plants in which the first product of photosynthesis is a 3 carbon acid compound called glycerate phosphate. Kwa mfano ni mazao kama wheat, rice, cotton, and sunflower. C4 plants are plants in which the first product of photosynthesis is a 4 carbon malic acid, 4 carbon compound called oxaloacetate. Unaona, oxaloacetate, yani OAA, such as maize, sorghum, sugarcane, and millet. Then finally you can finalize with the Kranz anatomy. Kranz anatomy, hii ni special structure of leaves in C4 plants. Ukizungumzia anatomy manake ni structure. It's the special structure of leaves in C4 plants, where the tissue equivalent to the sponge mesophyll cell is clustered in ring around the leaf veins outside the bundle sheet cells, or in the leaf structure in C4 plants, which consists of two photosynthetic cell types: bundle sheet cells that surround the vascular centers, and the mesophyll cells that in turn surround the bundle sheet cell. So the term Kranz anatomy manake ni ring in German, and the bundle sheet cell contain large chloroplasts with grana, whereas sponge mesophyll cells have few if any chloroplast and they lack grana. Kwa hiyo ni bundle sheet cell na mesoville cell ambazo zinatengeneza Kranz anatomy. Number 6, B Roman 2 tulihitajika factors favoring photorespiration. Photorespiration is favored by increase in temperature, increase in intensity of light, increase in the concentration of oxygen, and increase in the age of leaves. Effects of photorespiration, kwanza it decrease the yield of C3 plants in tropics. Na pili, plant may lose as much as half of the carbon dioxide fixed. Na tatu, they will be wasteful. There will be a wasteful loss of NAD pH2, yani reduced NAD katika huyo plant. Swali la sita, lo swala saba, with the aid of diagram explain the stages of Hatch-Slack pathway. Sijua kama kuna mtu ambaye hajui Hatch-Slack pathway ya C4 plant. Iko kwenye BS clear explained, lakini hata kwenye notes zangu clear explained. Hii ni swala la ku-explain to Hatch-Slack pathway, it's clear. Mention 5 significance of C4 plants over C3. Inawezekana wewe usijui 5, lakini in this question you are required to mention 5. B Roman 1, explain the importance of light dependent reaction of photosynthesis. Why there is importance of light dependent reaction, yani light reaction of photosynthesis, kuna umuhimu gani? Roman 2, Gabriel and Glory ate roasted meat and stiff porridge at their lunch. Account for the physiology of their meal along the alimentary canal. Physiology of their meal, that means you are required to explain the digestion of these meals. Actually, kwa swali hili, roasted meat ni protein and stiff porridge is carbohydrate. So you are required to explain the digestion of protein and digestion of carbohydrate along the alimentary canal. And I hope that's very easy for you to explain and to answer this question, digestion of carbohydrate and digestion of protein. This diagram na hili swali lote liko kwenye BS, kwa hiyo ni utaenda tu kulipitia hili swali hapa. Hii graph iko kwenye BS. Haya, figure below show the effect of increasing substrate concentration on the rate of a particular reaction in presence and absence of the enzyme. Unaona? Haya sasa, with reference to the figure above, explain the difference between the two graphs. Inaonyesha kabisa kwamba the rate of reaction is high when there is an enzyme. Hapo amesema explain the increasing the substrate concentration. Show the effect of increasing substrate concentration. And the rate of reaction increases with an increase in the substrate concentration when an enzyme is present. But without enzyme, the increase in substrate concentration, the increase in the rate of reaction is linear, tofauti na huko ambako inaonekana iko slant, yani the rate of reaction increase. Unaona? With reference to the figure above, explain the difference between haya. On the graph, draw two label curves to show the effect on the rate of enzyme catalysis reaction on the addition of a competitive inhibitor. Tukiongeza competitive inhibitor, nini kitatokea? Na B, tukiongeza non-competitive inhibitor. Kumbuka competitive inhibitor inafanya kazi with increase of concentration. Yani competitive inhibitor, the concentration of substrate increases, that means competitive inhibitor anafanyaje? Anakuwa the concentration of substrate increases, competitive inhibitor hawezi kufanya kazi. Wakati as concentration of substrate increases, non-competitive inhibitor anakuwa anafanyaje? Anaendelea kufanya kazi. So actually ni kwamba tukiongeza non-competitive inhibitor, we expect our graph itakuwa okay, let's clear that. We expect our graph itakuwa iko maeneo yale pale. Kwa nini? Kwa sababu a non-competitive inhibitor doesn't depend on the concentration. Doesn't depend on the concentration of substrate. Unaona, wakati ukiongeza competitive inhibitor, we expect our graph itakuwa hivi. Kwa nini? Kwa sababu a competitive inhibitor, it depends on the concentration of substrate. You see, so competitive inhibitor speed ya reaction ita change with substrate, lakini kwa non competitive inhibitor speed of reaction will not change with the substrate. Another day nikija na swali naweza nikaeleza vizuri au nikaeleza kwa ndani zaidi kuhusu hizo competitive inhibitor na nani competitive inhibitor in that case, but the concept you need to understand kwa hapo ni kwamba competitive inhibitor yenyewe kwa asili yake rate ya reaction ina change with substrate. Kwa nani competitive inhibitor haidependi kwenye substrate. Naona haya. From there, Roman 3, explain the effect of non-competitive inhibitor on the rate of enzyme activity. Effect of non-competitive inhibitor. Non-competitive inhibitor haitegemei enzyme concentration. Haitegemei substrate concentration. Sababu yenyewe ina inhibit enzyme regardless of the presence or absence of the substrate. Haya B, with examples explain 6 classes of enzyme. Najua ni moja kati ya swali ambalo uwezo kulikuta BS or understanding. Tuna class 6 za enzyme ambazo you need to know them, and the enzymes they are classified according to their functions. You need to know the 6 classes of enzymes. And then Lemon 2 explain 2 mechanisms of enzyme function. What are the 3 structural function of carbohydrate? Hivi vyote ni vitu ambavyo vipo kama kwenye notes zangu vipo. Kwa hiyo actually it's difficult kwa sababu kumfundisha mtu ambaye hana content nzuri ya theory inakuwa ni ngumu sana, lakini anyway nadhani you need to go and visit either my notes au notes nyingine zozote ili uwe na good knowledge in these parts, and then hence finally uweze kufanya vizuri katika hizo part. So thank you, and let I wish you nice studies for now. Let's meet in the coming session.